Hi everyone, welcome to lecture number 4. All right, so on this slide we just have a summary of the learning outcomes for lecture number 4. We're going to talk about types of data and we're going to revisit the idea of sampling and some really key concepts that have to do with sampling these types of data. I want to remind you that homework number 3 is due on Brightspace on Thursday, September 23rd by 4:30 p.m. And again, that's right before we'll have our Zoom meeting and we'll take up that homework on that day. Okay, so today, types of data, and then coming back to sampling. So there's two major types of data that we cover in this course, numerical data and categorical data. And they're just what they sound like. And each of those two major types has two subtypes within it. So we're going to start with numerical data or numerical variables. And these are just quantitative measurements. They're measurements that have magnitude. So they can be things like counts, lengths, percentages. So numerical data come in two kinds. We've got discrete numerical data. Those are measurements that can only take integer values or whole numbers, okay? So like number of friends that a person has, number of matings that a peacock gets, number of species in a community. Those can only be these whole numbers. So those are discrete numerical variables. Continuous numerical variables are real numbers. They're things that can take decimal values, like the height of a peacock, the length of a bird's tail, the body mass of an ant. So those are continuous numerical variables. Different types of data often entail different approaches to measurement and to statistics and visualization. And so that's why one of the core skills in this course is to be able to look at a data set or think about a measurement and tell what kind of data is this. Okay, so we're going to look at a few more examples of numerical data. This is a histogram showing you the distribution of a numerical variable. The variable here is the tail length of peacocks that I caught in my master's research. And so histograms, which we're going to cover again as we get into visualization in a couple of weeks, histograms are a type of graph that show you where most of the numerical values lie. So you can see this bell-shaped distribution. What it's telling us is that there are a lot of males with medium tail lengths and relatively few males with very short tails and relatively few males with very long tails. This bell-shaped distribution is a really common feature of many numerical variables. And so it's roughly symmetrical in this case. We see roughly kind of looks similarly shaped on the left and the right. So this is a normal distribution. It's symmetrical, it's bell-shaped. I just want to say histograms are a new graph for a lot of students. And so I just wanted to get into some of the features of it here. And so what we're seeing is these bars in a histogram are showing us on the y-axis how many individuals in our data set fall into these different bins. So the highest bar is the bin that's around, let's say, 104 to 106 centimeters. And the value on the y-axis of that highest bar, it's around, looks like around 41 or so. That tells us that there's 41 peacocks in our data set that had tail lengths between, let's say, 104 and 106 centimeters. Okay, so tail length is a continuous numerical variable. To reinforce what a histogram is, we have an explainer video on this in the course. And by the way, our explainer videos number 6 to 11 in the explainer video playlist are relevant to this lecture. You can find the link to the explainer video playlist on Brightspace, go to course home, scroll down to the section that says lectures, and there's a link to a playlist of videos that kind of, they're all like between 1 and 3 minutes, and they kind of teach these basic concepts of the course. They're really meant for you to study from. Here's a tip. The student that made the explainer videos, she used these really clever animations that are a really nice example of how to take good notes for your classes. They kind of map out the concepts, and I think she did a great job. So I think it's those videos give you a good kind of model for how to take good notes. Okay, here's another graph, another histogram showing you numerical data. Here we've got peacocks that I studied when I was a student, and we're plotting the distribution of the number of matings that these male peacocks got. So what kind of numerical variable is it? Is it discrete or is it continuous? Well, to answer that question, you have to think, can you have half a mating? Well, no, all these males, they either mated or they didn't, and some of them got lots and lots of matings, like some males got like 18 matings, let's say, but most of them, the histogram is telling us that most of the males mated between 0 and 5 times. Okay, so matings, number of matings is a discrete numerical variable. Okay, this distribution looks really different from the nice symmetrical bell-shaped one we saw for tail length. This one is non-normal. And there's different ways that distributions can be non-normal. In this case, we've got this long tail out to the right, a long tail out to the high end of the graph, of the histogram. And so we call that kind of a skew right skewed or positively skewed. That's something you'll need to know to be able to look at a distribution and identify it, describe it. Okay. Here's an example of what it would look like if it was left skewed. If we had the long tail out to the left, we'd call it left skewed or negatively skewed. Here's some real data on body mass. It's log transformed, so that's why we have some negative values, really small numbers that end up being negative when they're log transformed. But these are body mass distributions for both living taxa, living types of organisms, and extinct. So we have examples like on the top right there, we've got living mammals. On the bottom right, we've got extinct mammals. On the top left, we've got fish, and on the bottom left, we've got like, it looks like extinct flying things. So you can check out the paper that's referenced on the slide there. But I just wanted to show you there's kind of a range of distributions of numerical variables. Okay, body mass is what type? It's numerical. Is it discrete or continuous? It's continuous. And we've got an example of right skew data on the top right there. And then these extinct mammals have kind of a bimodal distribution. Bimodal means two peaks. We've got two kind of peaks to those to that distribution in panel H there. Okay, here we've got more data from the same study. On the top left, we've got body masses of a whole bunch of extinct dinosaurs, and this particular group of animals has a rather left-skewed body mass distribution. Right-skewed data are really common in biology. Left skew is actually more unusual, I'd say. And the reason right skew with a long tail out to the right is so common is that a lot of the things we measure in biology involve counts, and they often involve things that can't go below 0. So this histogram is an example of real data. This is counts of the number of individuals present from a bunch of different beetle species in the River Thames in England. Okay, so to do this study, they went to the river, they scooped out a bunch of stuff in nets, and then they kind of sorted it into species, and they counted how many individuals did we get of each of these beetle species. Okay, so question, is this discrete or continuous? So it's a numerical variable, but is it discrete numerical or continuous? So these counts of number of individuals, the variable on the x-axis. I want to show you what the data would look like because I find this a little bit hard to wrap my head around myself. I just made this up, but this is an example of what the spreadsheet of the data would look like. So we'd have a column for the species, and like these species, most of them don't live in the River Thames, but I just made this up, right? And then we've got our variable here, number of individuals in the river, right? So we've got, and what this graph is showing us and what the data show is that there's a small number of very common species. There's a small number of species with high numbers of individuals. And then there's many, many rare species, right? So, so if you look at the histogram over on the left, values down there mean that the species is rare, but there's like a lot of species that are rare and a few common species. Okay. Okay, so what kinds of data in biology are often left skewed? Well, there is one that's often left skewed, and that's lifespan data. This is real data on lifespan for golden retrievers. And you can just kind of see that there's kind of more of a longer tail out to the left. So lifespan is often left skewed. Okay, so that's numerical data. We have qualitative measures that also count as data, and those are called categorical variables. They don't have magnitude. They're describing membership in groups, describing qualitative characteristics of things. And there's two types of categorical variables, just like there were two types of numerical. And so we have nominal categorical variables and ordinal categorical variables. Okay, so what's the difference? Nominal means there's no inherent order to the categories. An example would be sex or gender, right? So we might have a variable, we're sexing beetles, we've got male and female beetles. There's no order to those two categories. An example of an ordinal variable, ordinals have to have a natural order to the categories. Maybe we have our beetles and we're classifying their life history stage, whether they're like a larva or an adult, for example. Okay, so there's a natural order to those stages. Maybe we have more than two life history stages, like larva, pupa, adult, but you can see how those have an order. Okay, and so categorical variables can differ in the number of categories. So we've got examples on here that might have two categories. That would be a binary variable, but there's other examples that might have like 10 categories. Here's some examples. I made up these data. These are bar graphs. We're going to get in a few weeks to types of visualizations, and we'll get into that some more. But you probably have seen bar graphs before. And so these made up data plot three different hypothetical categorical variables and show you the distribution for some imaginary biology students. So we could have gender, we could have hair color, we could have eye color. It's all made up, but you can see how we could have different numbers of categories here. Okay, so actually all of the variables in these examples are nominal categorical variables. There's no inherent order to the different hair color categories. Okay, here's an example of categorical data, real categorical data from this scientific paper on people's scores on a math test and where they were born in relation to their siblings. So if you have siblings, you might be the firstborn, the oldest in your family, the oldest child. You might be secondborn, thirdborn, fourthborn, etc. So this is not actually a bar graph. Well, actually, it is a bar graph, but this is showing you more than just the distribution. This is showing you a conclusion here. It's showing you the relationship between people's birth order and their average assessment on this math test. And the conclusion of this study is that firstborn children score higher at this math test than later born children. I don't know if other studies have kind of found that to be really robust, but there is quite a few studies showing that like firstborn children usually have an academic advantage over their later born siblings. Later born siblings probably have the advantage in other ways, and I'm sure there's lots of papers on that too. Okay, what I wanted you to notice about this example though is that birth order is an ordinal categorical variable, right? Because whether you're firstborn, secondborn, thirdborn, that's a category, but it's one that has an order. Okay, so just to summarize so far, we've got types of data. We've got our two big categories, and then each one has two subcategories. Really important point. Very often, something that you're measuring can be measured in more than one way. It could be one type of data, or it could be another, depending on how you do the measurement. And we'll see examples of that later on as you do your homework and throughout the course. In this course, we focus a lot on numerical and categorical data because these are the simplest and most common. And being able to identify the type is going to become important for types of visualizations and types of statistical analyses that we get to later in the course. So this is an example of how the material in this course really builds on itself, and having a good handle on identifying the type of data is going to be important now, and then you're going to need it later when we do other skills in the course. I want to do a little diversion here to introduce you to the fact that there are many other types of data out there. And so although numerical and categorical are the simplest, there's so many other kinds. And modern data analysis increasingly delve into these other more complicated types of data. And so I just wanted to give you a brief introduction to 4 of those types: spatial data, circular data, network data, and genetic sequence data. Because if you go on in biology, if you go on in data analysis, you're going to encounter these kinds of data. And when you read papers, you will as well. Okay, so that's just summarizing what I just said. Okay, spatial data. Here's an example of data from a real study where they put these GPS trackers on cats, like house cats that people would let outside. And so we've got these traces in 2-dimensional space, these latitude and longitude coordinates tracing a path through space of where these house cats went in the neighborhood whenever they were being let out. Okay, so there are numbers here. We've got latitude coordinates, longitude coordinates, but it's more complex than a simple numerical variable, right? We've got like a position in 2D space. So that requires its own types of analyses to deal with. And we don't get into those types of analyses in this course, but I wanted you to be aware that there's more to kind of the diversity of data than just simple numerical data. Here's an example, another type, circular data. This is real data on monarch butterflies and how they're orienting during their migration, their fall migration period. Okay, so it's from a study where the investigators wanted to understand how these tiny insects use cues like the sun to know where to go in their migration. So the data, you can see them plotted on these kind of like compass plots, right? So we've got north, south, east, and west. We've got these data points. These are like histograms, but they're like histograms for circular data. So they're showing you the distribution of where the monarchs oriented themselves, where they tried to fly during these experiments. So circular data is special because it's not on a linear scale, it's on a circular scale. So like, as we go like northwest, we're getting increasingly close back to north and northeast and so on. So you can't analyze it the same way as you do linear numerical variables. Okay, here's another example of type of data that's increasingly important: network data. So in the graph we're looking at here, it's from a real study. It's showing you some human social networks. And so each of the little dots here is a person that they studied, and the researchers here asked people if they were smokers or not, and they also mapped out who each person was connected to in terms of their friendships in this community. And then at the bottom, you can see some kind of zoomed in details from these social networks. Okay, so this is not like a simple linear thing. We've got actually 2 kinds of data here, right? We've got for each person, we've got this nominal categorical variable, were they a smoker or not? But we've also got like, who were they connected to in this web, in this social network? And what this study argued was that people tended to be connected to other people who were similar to them in terms of whether or not they smoked. And so that's really interesting and raises other questions like, is smoking behavior contagious? Or do people who are members of one of these groups, either smoking or non-smoking, do they tend to be more attracted to friends that are more similar to them? Well, it's been proposed that a whole bunch of behaviors like kindness and cooperation and healthy behaviors like exercising might be contagious and might spread in a social network. Okay, so these types of data can have really interesting nonlinear effects because something that starts with one person can easily spread to a whole bunch of people in a network, right? And so those kinds of network data are really important, especially during something like the COVID pandemic. Now, social networks are not the only kind of networks in biology. Biochemistry is full of networks. Food webs in ecology are networks. You can go beyond biology. There's networks everywhere. There's like traffic networks in cities. There's networks that map how airplanes connect different places in the world. It's everywhere. Okay, last example of fancy data, genetic sequence data. This is hugely important nowadays. You should go on and take Carleton courses in bioinformatics, molecular genetics. It's a hugely valuable type of data in industry today. Okay, so all of these types of data, these fancier types of data, can be used to derive simpler types of data as well, right? So we could take some of these fancy types and derive numerical variables. You can imagine how the spatial data on where those cats went can be used to derive a numerical variable of distance traveled. Okay, so practice. Here's some made up data on an experiment where we were interested in how the breakfast somebody eats influences their running speed and also including their caffeination and what they ate. So it's a bar graph with invented data. There's two variables here. There's a variable running speed on the y-axis, and then there's the variable breakfast on the x-axis. I want you to practice what type of data are each of those two variables, right? And so there's 4 types. It could be either numerical, discrete, numerical, continuous, categorical, nominal, categorical, ordinal. Okay, here's another example. Practice this as well, and we'll take up the answers on Zoom. What type of data is mortality rate on the y-axis? We've seen this graph before. It's showing us mortality rate of humans and cats that fell from these big buildings. Mortality rate on the y-axis, and then number of stories fallen on the x-axis. And there's actually a third variable here, species, right? We've got humans and we've got cats. So what type of data are each of those 3 variables? All right, so at this point, I want to come back to sampling. This is a really key concept of this whole course. And we saw in the previous lecture how sampling can be really powerful. It can help us predict an election by interviewing just a subset of all of the voters. But if we don't do it properly, it can have some surprising consequences. And I want to tell you about a really fascinating example that turns out to be a case of sampling. And it starts with some sports legends. And so this example is called the Sports Illustrated Curse. Okay, so in 2015, the Toronto Blue Jays appeared on the cover of Sports Illustrated magazine for the first time in many years, and they got this like nickname that I guess means that they're really doing well. And after that issue of Sports Illustrated came out, the Blue Jays went on to lose a really important series to some other team. So that was not a very good prediction on the part of the magazine. And then something similar happened again pretty recently. In 2019, the Golden State Warriors were featured on the cover, touting how great they were, and you probably know how that turned out for them. They lost to Toronto that year. Okay, so the Sports Illustrated jinx or curse is this urban legend that states that individuals or teams that appear on the magazine will subsequently have some really bad luck. And if you go on Wikipedia and look this up, this is just like a screenshot of a small number of the examples in the Wikipedia article on this. There's I think there's got to be hundreds of them, like in Olympians, college and high school athletes, all of these like up and comers, they make it onto the cover of the magazine, and then some horror, they do really badly, or they get a big injury or something bad befalls them. So the Wikipedia page is 15 pages long on this curse. So there's a lot of examples. So what's going on? Well, it has to do with sampling. If an athlete is on a winning streak, like the ones on the cover are the ones on these winning streaks, it's a lot of skill, but there's also some chance to it. There's also some random chance to being able to do well, right? And so there's some luck that has to do with it. And eventually, random chance is going to go the other way, right? With things like injuries. And sometimes you lose just due to bad luck. And so this phenomenon is called regression to the mean, and it's sometimes also called reversion to mediocrity. And what it says, what this principle says, is that if a subject is extreme at one measurement, on average, it's going to tend to be closer to the mean at another measurement. And this phenomenon goes in both directions. It can go forward or backward in chronological time. It can apply to both types of extremes, high extremes or low extremes. So I'm going to try to flesh that out for you in the next few slides. So here's a diagram to try to show you this concept. So imagine we have these 3 professional athletes. They're all much higher performing than we would be, and they each have their kind of histogram of their distribution of performance values. So these are 3 histograms like tilted on their side, right? So the black bar is like the mean performance for each of these athletes. And sometimes they have their good days and they have their bad days. Okay, so there's a threshold to appear on the cover of Sports Illustrated, right? Like they're not going to be on the cover unless they just recently did some really good stuff, like at the peak of their personal performance. Well, guess what? On average, when you look at them at some other time, they're going to be doing worse than that peak performance. So that's regression to the mean, and that explains the Sports Illustrated curse. It's not some magic thing. It's just a feature of sampling. Okay, so the lesson here: don't focus on extreme values. If you're doing a scientific study, don't limit your data to extreme values, because you'll run into some paradoxical conclusions if you do that. The extreme values are not a random sample. They're not a representative subset of the original set. Okay, so if we just look at athletes right after they've been on Sports Illustrated, it's not a representative subset of times in their performance history. So if we limit our data to extreme values, we're going to have an overrepresentation of cases that are unusually extreme in that direction. And another way to say this is, if you measure me at my worst, on average, I'm going to score higher at other times. And vice versa, if you measure me at my best, on average, I'm going to score lower at other times. By the way, I apologize for the noise. I don't know if you can hear it in this recording, but there's like some tree chopping that's going on outside my house right now. So I hope it's not too loud on the video. Okay, here's an example where regression to the mean matters a lot. Okay, this is the Scared Straight program, which was pioneered in the United States, and it was adopted in other countries around the world. And what the program was they took juvenile offenders and they took them on tours of real adult prisons. The juveniles met inmates who told them about prison life and said, like, this is how bad it's going to get if you don't stop your criminal ways. So the question is, did that program work? Well, a study was done in New Orleans, and they found that after participating in Scared Straight, these young people were arrested less often than they were before, right? So after they were put through Scared Straight, they were not arrested as often as they were before Scared Straight. Okay, so maybe Scared Straight works. But we have to think about regression to the mean here. We're taking a group of juvenile offenders, we're selecting these people at kind of their worst behaving moments, right? So we're taking them right after they've been convicted of a juvenile offense. And the chances are, if we're selecting on a group kind of at their worst, chances are those kids might behave a little bit better, or maybe get caught a little bit less next year just by chance alone. So the question is, how should we measure the effect of Scared Straight, right? What can we do? Should we compare juvenile offenders' outcomes to their own bad behavior before they were treated with the Scared Straight program? Or should we compare their outcomes to other juvenile offenders who are not exposed to Scared Straight? Okay, this is a diagram to kind of try to show you the concept again, the same idea as the Sports Illustrated course. We've got these young people, they've got kind of distribution of lawfulness, and there's the kind of each person has their own mean. Different people might actually have different means. In this case, they all are shown the same, and they've got days when they're very well behaved, and they've got days when they're very poorly behaved, but most of the time they behave around their own personal average value. If we select like when we look at the people who were put through Scared Straight, we're kind of looking at these people at their worst, right after they got convicted of these offenses. So it's no wonder that when you measured them again a year later, that on average they'd be more lawful. And really interestingly, after the New Orleans study, there was a study looking at Scared Straight, where instead of comparing these offenders to themselves, they compared them to another group of young people who were also offenders, but who were not put through Scared Straight. And what that study found was that the kids who were put through Scared Straight were actually more likely to commit crimes the next year as compared to the offenders who were not put through Scared Straight. So Scared Straight seemed to have the opposite effect of what it was designed for. So that's an example where we really want to know that conclusion. And in this case, we need a control group that is similar to the group of people put through Scared Straight in every respect, except that the kids in the control group are not put through Scared Straight. Okay, so when we're thinking about these things, we're really thinking about variation and where it comes from. And I'm going to define replication as just meaning whenever you repeat a measurement. So you can repeat measurements on the same individuals, like an athlete or a young offender. So we can kind of imagine measuring this athlete on different days. We've got replication. We've also got replication in terms of every time we sample from a population, that's a replicate from that population. And if you think about one measurement to the next being different, where do those differences come from? This is a major question in biology. Let's think of some potential sources. Let's think of the athlete. Why might they differ from one day to the next in terms of their running performance, for example? Well, we've got, genetics. Okay, one athlete doesn't change their genetics, so maybe that's not a good example for thinking about one athlete, but that's a major thing in biology. Some runners might be better than others due to genetics. We've got experience, prior training. That might explain differences among different runners, but also it might explain differences why one person might improve over time. We've also got time of day, season, environmental conditions, weather, what they ate, how much caffeine they had. We've also got features like measurement error, right? So maybe our instruments, maybe some of the variation in the numbers we get are due to error in our instruments. And then we've got just random chance, luck. It's hard to separate that from environmental conditions sometimes, but you can imagine how, if somebody almost got run over by a car on their way to their race that day, that's a random chance event, but that might impact kind of becomes an environmental effect that might impact their performance at the race. Okay, so we can kind of think of biological sources and chance sources, and sometimes it's hard to discern the difference between these two things. Okay, let's think about how does chance affect estimates that are derived from sampling. So now let's think about, okay, we're drawing a sample from a population. That means chance influences which individuals we get, right? Well, whenever we're sampling, we're going to get differences from one sample to the next. And we're also going to get differences between the sample and the population. And those differences that are just due to sampling, just due to who you happen to sample, those are random, right? They arise simply by chance. You experienced this yourself in homework number 2. Homework number 2 had this simulation sampling heights from a population. Each time you click the button and draw a new sample, it differed slightly from the last one. And it also differed from the source population. Okay, now coming back to this diagram of estimation, I'm going to add on to it. I'm going to build it up. So last time we looked at biased sampling. So that creates systematic error. Now I want to add in random chance sources of error. Okay, so now we've got this like two-dimensional diagram here. In the vertical axis, we've got precision. Random error, including sampling error, that affects precision, right? So if you look at like the left column of this diagram, and so the top left, we've got very precise estimates. Our estimates are tightly clustered together. In the lower left, the estimates are more spread out. So from one estimate to the next, we've got bigger variation. Okay, that's less precision, right? Like tightly clustered estimates, precise. Spread out, not precise. If we look at the right column, top right, they're tightly clustered, they're precise, but they're way off the true value of the target. So they're precise, but they're really biased. And that's, we'll talk about on Zoom how that can happen with sampling. On the lower right, we've got bias. The estimates are way pushed off the true value in the population, but also they're not precise. They're like spread out. So like that's the lower right example there is like not good estimates on both counts, right? So where we want to be is in the top left. We want estimates that are unbiased and that are precise. I just want to say that we're going to look at these concepts in some of our first examples, thinking about numerical estimates. But these concepts apply to any type of data you could be sampling, whether it's numerical or categorical data. Okay, so just to summarize again, differences between samples or between the samples and the population that arise just by chance, these random differences, that's going to affect precision. And that is a new thing that we've learned, and we're building on to what we covered last time, where bias refers to systematic differences between the samples and the population. And we in statistics, in kind of measurement theory, the word accuracy is often used to contrast with precision, but that gets confusing because accuracy has kind of like a meaning in everyday speech. So that's why I've tried to stick with talking about bias, unbiased. But you'll sometimes see it being referred to as like bias means not accurate and unbiased means accurate. So that's my caveat about language. Okay, I already said that. All right, so practice that we'll take up on Zoom, go back to the simulation from homework number 2, where you're drawing heights from a population and you've got this like histogram. Every time you drew a sample, the histogram of your sample heights looked a bit different. Play around with that. What do you think is the best way to reduce the effect of random chance on your sample? In other words, what's the best way to improve precision of your estimates? Okay, so we'll take that up when we meet on Zoom. Okay, here's a different example to kind of illustrate some of these concepts again. This is a little study that I did with the students in this course back in 2019. I had 100 students tell me their guesses for how old these celebrities, these 4 famous people, were. And then I kind of graphed the results. Okay, so these are histograms for the age guesses by Biology 1105 students for these 4 different famous people. Okay, so each student provided one guess for a given person, and then you can graph the histogram of the numerical variable of those guesses, right? Okay, so here in the red are the actual ages of those 4 people. So look, this is really interesting. Right away you can see how the distribution of guesses might be well aligned, might be centered on the actual age of that celebrity, or it might be pushed off of the actual age of that celebrity. Okay, so I calculated the error or the difference between each guess and the celebrity's actual age. And in the middle column of this slide, it's just showing you those error calculations. So in the middle column, each point is a student's guess. So like you can see how like on the top left, a whole bunch of students thought Keanu Reeves was 30 years old. And then the horizontal lines are calculating the difference, right? So it could be a positive difference or a negative difference. If the student thought the celebrity was younger than they actually are, it would be a negative number. So I took the guess minus the celebrity's true age, and that was my error. And then on the far right, I'm graphing those the distribution of those errors. Okay, so right away you can see if the students were guessing bang on average, like that distribution could be center of error, it could be centered on 0, or it could be pushed off. So let's zoom in on sources of error for Keanu Reeves' age guesses. So orange is the average error for the class, and if the error was 0, that would mean like a correct guess. And so we can draw a couple of illustrations here. We can look at the kind of spread in this distribution of estimates. So in blue, like how wide is that distribution of estimates? And we can also look at the difference from 0 to the average of the class. Okay, so those are illustrating these two concepts of bias and precision, right? So in blue, how broad that distribution is, that's telling us how consistent are the estimates of students in the class, right? So the wider it is, the less precise students are as age guessers. The tighter together that blue line is, or the tighter students' guesses are, the more precise. In green, we've got our bias, okay? So students think Keanu Reeves is younger than he really is, right? So we see this like bias towards guessing that he's significantly younger, like 10 years younger on average than his true age. So that's a good thing for him. But overall, students were biased at guessing his age. And so that indicates that there's systematic error going into these estimates. All right, so that's it for this lecture. Remember, homework number 3 is going to be due Thursday, September 23 by 4:30 on Ottawa time, and you can submit homework number 3 on Brightspace.